everybody, I'm Megan Ross. I'm Coordinator of Student Development and Student Programming in the College of Agriculture. And thank you for joining us for our Work seminar. And we are here at HudsonAlpha. We're going to be spending some time with Alex and a few of his friends, learning more about what they do here. Tell us about HudsonAlpha.
So this is HudsonAlpha Institute for Biotechnology. This is a nonprofit research institute that studies the genomes of all living things, from humans to animals and to plants.
Cool. Let's take a look inside.
Sounds great. Let me take you in. What is biotechnology? Biotechnology is a pretty broad term. I think it's almost a 100-year-old word. If you were to ask me right now, I think biotechnology is the use and study of living systems to develop and produce new technologies that can improve plant and animal biology. HudsonAlpha was initially founded as a human genetics research institute, and the idea was to use this incredible new technology of DNA sequencing to study the genomes of many human beings, to understand the genes that might underlie different diseases or cancers or neurodegenerative diseases. And since that initial founding almost 20 years ago, HudsonAlpha has expanded not just in humans, but all sorts of animals and to a huge diversity of plants, which is part of the reason why I am here.
Yeah, so the role of biotechnology in agriculture right now is very intertwined. If we talk about just genetics, so if we don't talk about all the other ways that you can integrate bioengineering and biotechnology into agriculture, just genetics. Traditional breeding is very simple. You identify lines that are good, and you use this idea of transgressive segregation, which means that if you have two parents, the progeny of those parents, there are some that are better than either parent. So for instance, LeBron James is much better basketball than his mother or his father. That's what breeders are looking for, okay? And so they do that by this very long, careful, beautiful process where they make the crosses, they put all the kids out in the field, and they walk around, take notes, and say, this is the best kid. This is the LeBron James. And then they test it again and again and again, and they release that as a variety. So with biotechnology, we can identify some of those traits that we know will make LeBron James, or maybe it makes Kevin Durant, right? So something like that. And we can select for that in the lab.
So what are all these?
Well, this is really kind of the heart of the expertise here at HudsonAlpha. These are DNA sequencers. Each of these machines, the NovaSeq 6000, are short-read sequencers, meaning we can produce almost 3 trillion nucleotides of data per machine over about a 2-day period. And we have 8 just in this room alone. As well, in the back, you can see we have long-read sequencers. Those make huge pieces of DNA sequence, like 20,000 nucleotides or so. And that enables us to put plant genomes together or to resequence tons of human genomes. This place can produce thousands of human genomes each year.
Oh, wow. So these used every day? Like, how often are they in use?
These are always running. There's always something on them.
Cool.
So a really unique aspect of HudsonAlpha is that there are 40 biotech companies on campus, many of which are in this building right here. So that allows biotech companies to interact with faculty laboratories like mine, so that Auburn students in the College of Agriculture or beyond get to interact with all kinds of people, from government science to biotech startup life to mature companies and academic labs like mine.
Wow. So they get a broad experience.
Yes, and get to network broadly because there's almost 1,000 people, if not more, on campus right now.
Oh, wow.
So how did I end up at a place like Auburn and HudsonAlpha? There's a really incredible partnership between academic universities and nonprofits, and I think there's no better way to exemplify that than what Auburn and HudsonAlpha have done with a position like mine. So Auburn University has long-standing expertise in plant agronomy, plant agriculture, nearly a 150-year history of growing plants on Alabama soil and deeply and emotionally understanding how plant biology works in this state. And then you think about a place like HudsonAlpha, which is a nonprofit biotechnology institute, which has immense global expertise in the genomes of diverse plants and animals. So in many ways, I was attracted to this position because it combines local expertise of two very powerful research institutions that together we can become greater than the sum of their parts.
So tell me about the plant side of HudsonAlpha.
So there are 5 faculty laboratories here at HudsonAlpha that study plants. There's my laboratory, joint between Auburn and HudsonAlpha, that studies plant reproduction and plant evolution. There's Jeremy Schmutz's lab, who studies computational genomics and comparative genomics in plants. Jane Grimwood's laboratory, who runs the HudsonAlpha Genome Sequencing Center here. There's Josh Clevenger's laboratory, who studies molecular breeding in plants, particularly with a focus on improvement in peanut. And lastly, Kangshita Swaminathan's lab studies functional genomics in plants and understanding what genes do.
All right, cool. Cool. Can we check out some of those labs?
Yeah, let me take you.
So tell us about your lab.
Yeah, welcome to laboratory. My laboratory and Josh Clevenger share this space. This is where we generate all of the molecular work that needs to be done to fill up those DNA sequencers. And someone who's been really instrumental to our laboratory is our lab manager. This is Haley Hale.
Hi, yes, I'm Haley Hale. I'm the lab manager for both the Harkis lab and the Clevenger lab. Yeah, I just am involved in getting the lab set up, purchasing and ordering, but most importantly, taking like plant tissue and getting it ready for sequencing. So extracting the DNA, building libraries, and then taking it over to the sequencer. And that's when Alex and Sarah come into play. But yeah, this is my realm.
And that's a really awesome part about being at this institute, is because there's so much historic knowledge of excellent molecular biology. And so Haley can extract DNA in our laboratory right here and walk it not 50 steps away to go talk to some of the people who were involved with the first human genome sequencing project and gain their expertise too. So we're all learning from each other, and we're all building on a really excellent history of plant genomics expertise.
Yeah, and every day is a learning process. I mean, especially when you're working with labs that we work with peanut tissue, we work with trilliums, duckweeds, asparagus, like Mirica. Every plant is different, and so it's a bunch of troubleshooting, but it's a giant puzzle that's very exciting to work with.
I like that you say that. A lot of people at this institute study one species, humans, and then you come to our laboratory and we study all kinds of species. There is no limit to what we can work on. And that's the fun part about kind of being in a laboratory like this, is as long as we can get DNA, we can make a genome and we can start telling stories about how that plant has changed over time. My laboratory studies how plants have evolved to reproduce in so many diverse ways. Roughly 90% of plants that make flowers make hermaphroditic flowers that have both male and female parts inside of every single flower. I think that's an amazing adaptation. In many ways, these plants can often self-pollinate and always produce a seed. But of the several 100,000 species of plants that make flowers, maybe 10% of them do things a little bit differently. In many cases, they've evolved separate sexes, so that some plant species have individuals that only produce male flowers or some that only produce female flowers. This has happened multiple independent times. Hundreds or thousands of independent times have plants evolved away from this ancestral hermaphrodite flower. What my laboratory does is we study all of these independent events that, where plants have changed the way that they sexually reproduce. And we find the genes that control whether a plant is male or female or hermaphrodite or neuter or something in between. And that's a very basic science question. It leverages classical botany, evolutionary genomics, and our ability to compare multiple genomes against each other. But in many ways, my laboratory studies the past. The first green thing evolved about 1 billion years ago. Plants moving onto the land maybe 500 million years ago. The first flower evolved about 130 million years ago. Humans have only been farming for 12,000 years as a species. So my laboratory uses genomics to look at that incredible rich history, to find all of these ways over time, over hundreds of millions of years, where plants have changed the ways that they reproduce. And once you do that, once you map out all the genetic changes that can control whether a plant makes one flower or another, you can start to engineer that into crops to improve the future. So in many ways, my laboratory uses the past to hopefully improve the future, particularly with hybrid crop breeding. How do we control how two plants reproduce with each other?
Yeah, so I am a scientist that did not come to this in a linear way. So I love music and I play musical instruments. And so when I was younger, I wanted to be a rock star. That did not work very well. And so in my mid-20s, I went back to school. When I went back to school, I fell in love with conservation, plant conservation. I was really excited about the woods and conserving natural diversity, and realized that I could do that by working on genetic improvement of plants and breeding, so that we could have genetics in the growers' hands that they could grow and have less inputs, less fungicides, less pesticides, to help with that sort of stewardship with the natural land around them. So I went to Ohio State and I studied molecular genetics in tomato. I studied how tomatoes sort of got the shapes that they have. So long shapes, round shapes, weird pumpkinish shapes. And then from there, I went to University of Georgia and studied peanuts, particularly the genome of peanuts and also aflatoxin contamination in peanuts. From there, I worked for Mars Wrigley, a candy company, as a scientist for a food company, and sort of learning about global supply chains and how international companies that do billions in sales operate around the world, and how that interacts with crops, with plants, with growers, with origins where they buy their chocolate or they buy their peanuts or their other nuts that go in their products, and how complex how complex that is, and how sort of the issues that farmers face around the world are very different and very serious, and how we can combat them. And so from there, I came to HudsonAlpha as a faculty investigator to study translational genomics, which basically means using genomics in an applied context for direct genetic improvement for breeding applications. So instead of having genomicists that do genomics and then breeders that do breeding, I bridge that gap and do both things. Here at HudsonAlpha, I'm focused on peanut for a couple reasons. One reason is I love peanuts. They are an incredibly healthy food that is very cheap. And I'm in love with that because it means that people in poor areas, people in food deserts, can go to the corner shop and go to the 7-Eleven and afford a pack of peanuts and get a health benefit that people in more affluent areas maybe are getting from almonds. And so it's a really, really interesting food in that way. It's loaded with vitamins and minerals, healthy fats, and it's very, very successful at treating malnutrition in kids because it's good for them and they like the taste of it. So there's a lot of really great aspects of peanuts. Peanuts are also very important in the South. They're very important in Alabama, and I know a lot about them. It's also what we call an orphan crop. And so that's sort of silly to say about peanuts because actually they're worldwide very important. They're one of the most important oilseed crops. But in terms of public funding, we call them an orphan crop because most of the research is funded by private industry. So you don't see a lot of governmental grants doing research on peanuts. So when you put all those things together, plus the very successful breeding program now, the new nascent breeding program at Auburn, with their first release that I think is a fantastic cultivar. Knowing that peanuts don't get a lot of public funding, and being here in Alabama, it seems like a perfect way to utilize the strengths of HudsonAlpha, the genomic strength of HudsonAlpha, to really do cutting edge research on a crop that otherwise wouldn't have that. And so that's what we do. When I did my PhD, I was heavily involved with the Peanut Genome Project, which was an industry-funded project that was wildly successful to sequence the peanut genome. Now we think of that, for instance, in the past few months, I've sequenced 10 peanut genomes. But at that time, it was when I started, the technology was to a place where we didn't think we'd actually be able to do it until the advent of what we call long-read sequencing gave us the ability to sequence that first peanut genome. And that was done in collaboration with HudsonAlpha. And so what can we do with that information? So after the genome was assembled and published, what we now could do was develop better marker sets, so better ways of genotyping peanuts. And that feeds into trait discovery, that we can identify genes, identify markers to select for those genes in a breeding context. And all of those things together allow us to improve peanuts faster. So that's what we do.
I'm a plant biologist, and I guess my interest in plant biology was one of the last things actually that I thought about working on. And so slowly, from the macroscopic level of just being fascinated by the things plants do, I kind of moved into the DNA level and genomics and what genomics can tell us about genes. And then from there, I learned that, by studying genomics, you get to a lot of hypotheses, and you wanted to understand what the genes actually do. And that took me into biotechnology and gene editing. So here in my lab, what we focus on is problems in agriculture that relate to sustainability. So for example, we've bred our crops in a way where we have a lot of fertilizer, we can throw a lot of fertilizer, and all plants do really well when they're fed well. And so they produce a lot of yield. But then that has an environmental impact. And so we've got green waters and situations arising from overuse of things like nitrogen. And so to really understand how the plant uses nitrogen, and this is an important thing to understand how we can find plants that actually use it well and how little we need to actually add to make it yield well. And so a lot of what we do here is the basic biology. So for example, we have an NSF funded project that looks at sorghum, which is a grain crop, and it looks at how does it actually respond to nitrogen. So this is a collaboration with the University of Nebraska, and what we do is really use techniques like automated phenotyping, transcriptomics, which is basically looking at gene expression. And we combine the two to try and understand how does the plant actually respond to different levels of nitrogen. So we gave it 5 different levels of nitrogen and looked at how the plant responds. What we do at the end is we get a lot of data that from that data we can kind of extract a hypothesis that says these genes appear to be very responsive to nitrogen. And so the next question is, what do they do in this process? To get to that, the lab uses a technology like biotechnology, where it is basically plant transformation. So you can manipulate the expression of that gene, or you can take the expression of the gene completely away using gene editing with new technologies like CRISPR-Cas9, which basically, you can make the plant just have a mutation in your gene of interest using these technologies, which would then take away that gene function, and then do the same experiment all over again to determine how removing that particular gene function influenced the phenotype that you were looking at. So how does it respond to N? Does it change anything or not? What else does it have an effect on? Questions like that. In the same way, you can actually increase the expression of a gene by overexpressing it, by putting something called a transfer DNA into the plant, which actually expresses this gene at a higher level and asks the same question. So if I have too much of it, what happens? If I have none of it, what happens? What happens in the wild type where there is some of it, right? And so now you have this comparison which can help you get at that gene function and determine whether that gene is important for nitrogen. So that's kind of what we do.
There is a real power with an R1 land-grant institute like Auburn University connecting to a nonprofit biotech institute like HudsonAlpha. It opens up incredible doors for students to explore the incredibly broad, diverse, and growing field of biotechnology, plants, animals, and humans. So how are we bringing plant biotechnology directly into the hands of students at Auburn University? One really exciting example that I'm particularly thrilled about is that I teach a plant genomics course at Auburn, where students actually take raw data from the sequencers here at HudsonAlpha, and they assemble, annotate, and publish a plant genome as part of a course. And in our first year, we're assembling the tumors oak genome from Auburn's downtown campus. And the idea is that students in my course, over the course of a semester, will be putting together the chromosomes of the tumors oak genome. They'll be finding the genes on those chromosomes, and together as a class, we will write a peer-reviewed manuscript where every student will be a co-author on a paper that will be submitted to a real journal. And the idea is that by doing this, we increase access and we reduce barriers to the field of STEM, and particularly to plant biotechnology. My dream is that by offering courses like this, where we put biotech into the hands of students, we can increase the representation of all kinds of people into the field of STEM.
I would say that if you're interested in research, try your hand at various different types of research. You never know what really clicks and what your passion is. At least that comes from my own experience, where you really had to, try a whole bunch of different things. I had to try a bunch of different things to finally figure out what I could wake up every day and be thrilled to go into work with, right? So I think as a student, working at a university, there are lots of options for you. So go to a lab and figure out if you like doing it. And if you don't, it's not time wasted because you've just eliminated one particular path, right? And then you find the next one, which you might enjoy better. And so here in summer, we have something called the BioTrain program, where a lot of students apply for this program, and it's a short course where you get to work in labs or in any place, any of the departments at HudsonAlpha. And I actually have a student, Charis, who's from Auburn University, who is spending her summer here, and maybe she can tell you a little bit more about the kinds of things that she does, which is a little bit different from the things that I actually described.
So my name is Charis Harrison. I am going into my junior year at Auburn, and this summer I'm a BioTrain intern at HudsonAlpha in the Swaminathan Lab, working on plant genomics, mainly phenotyping of miscanthus. As far as what got me interested in undergraduate research, I honestly can't really remember. It's just something that I've always been interested in. I've always really admired the work of people that are discovering new things and creating new products and things like that. So as soon as I got to Auburn, I knew that we were an R1 research institution, and I knew that that was going to be where all of my research opportunities lied. So coming into my sophomore fall, after everything shut down because of COVID, Dr. Noel came and presented to the microbiology club and said that he had openings in his lab. So I applied, interviewed with him, got accepted into the lab, and it's just been a really great experience so far. What I like about HudsonAlpha a lot is that it's a very collaborative environment. I'm working really closely with the senior scientists and the PI in this lab, but also getting to see what's going on in the other plant labs on campus, like the Josh Clevenger lab and the Alex Harkis lab. In addition, I'm getting to see and work with a lot of equipment that I had never even known existed before, and it's giving me really good exposure to what non-academic research is like.
So in terms of how I would give advice to someone who is interested in agricultural research as a young person, and when I talk to young people, my career has been very interesting and it's taken lots of turns. When I was doing my PhD, I thought I would try and get a postdoc with a famous person, and then try and get a faculty position and be a professor. And that's what I thought I wanted to do. Someone from the candy company said, "We want you to work for us, and this is what we want you to do." And I said, "This is pretty interesting," because it gave me the ability to have this sort of global effect, right? So I could affect things globally instead of locally. And so that was really interesting to me. And during that process, what I learned is that, number one, this individual noticed me because there were leadership opportunities I did not turn down. So at public meetings, when people asked me to do things as a young person, I didn't say, no, I'm kind of shy or I'm scared of that. No, I took that chance and I did it. And then I was noticed. So the first thing is you never turn down an opportunity to make an impression. That's the biggest thing, because there's always somebody you don't know that is watching you. And I have this great picture that was taken from that meeting of me sitting a few seats away from this gentleman who's my mentor from Mars. But we did not know each other until that meeting, when he saw me speak and he said, "I'm impressed with this person." And you're never going to know who that person is or where they will be that might notice you and really take your life in a different direction. That's number one. Number two, always keep your mind open, because you never know what opportunities are out there. There's so many interesting things you could do. There are people at Mars Wrigley that research how to have gum dissolve faster, but not dissolve fast enough because you want people to chew it, but dissolve fast enough so that they don't take up space in landfills. Who would have thought when they're doing a biology degree or a chemistry degree that might be something they're working on?
So how do you put together those plant genomes?
Plant genomes are really difficult in many cases. Imagine putting together a plant genome kind of like putting together a puzzle that doesn't have a box. You don't know what it's supposed to look like. 5% of the pieces are missing, a bunch of the pieces have been chewed on by a dog or something, and then when you start to lay out all the pieces, there's no edges, there's no corners, and they all look like Dalmatian spots. How do you put together a complex puzzle like that? Well, we can teach computers in many ways of how to put those complicated puzzles together, at least to a point. One solution is to make bigger puzzle pieces. And to put that in the context of what we do here at Hudson Alpha, we use DNA sequencing machines that can take huge pieces of DNA and actually read them, kind of like speed read them, to convert DNA that is a clear, colorless liquid in a tube into ATCG, into these stretches of DNA sequence reads, puzzle pieces.
Yeah.
And if we make huge puzzle pieces of these 4 nucleotides, then we can teach computers how to overlap them, put them together, and assemble chromosomes from end to end.
So you can see what the plant looks like on the inside. Complete picture.
We can see what the plant looks like on the inside. That's exactly right. And that whole process starts by extracting DNA out of a plant, like from a leaf, for instance. That's all it takes. And once we get that DNA, then we can put it into these DNA sequencing machines to make these big puzzle pieces.
Cool. Well, can we take a look at one of those?
Absolutely. So these are the cutting edge of DNA sequencing technology for plants. These are PacBio Sequel II DNA sequencers. These allow us to generate those huge puzzle pieces of nucleotide sequence that we talked about that make putting together these complex puzzles or plant genomes much, much easier. So we can extract DNA from a leaf in my laboratory just right across the hall and immediately bring it over to this kind of machine and generate genomic data for any plant species. And that's the really amazing part, is that this machine doesn't care what you put into it. Any plant now is able to become a model system in our world because we can generate an ultra high quality reference genome for it.
Oh, wow. So how long does it take if you were to put in a leaf? How long would it take to come up with that?
about 48 hours to get raw data, and then after that, we can put together our first draft of a genome in a couple days.
Oh, wow.
So this is partly why we're starting to bring this technology into the hands of students at Auburn. Because here at HudsonAlpha, we have access to so many of these sequencers, we can produce data very, very quickly, and we have the computational resources to put genomes together very quickly.
That's awesome. Cool. So these are just these are the early plants, the first plants.
Yes, these are a bunch of the plant species that we've sequenced here at HudsonAlpha. And what you might not realize is that this institute is a global leader in the production of ultra high quality plant reference genomes, meaning that this place has produced more than 200 complete plant reference genomes that exist that are publicly available for anyone to use. That's more than I think any other place in the world has ever produced. Where do I see the field of biotechnology in 20 years? I think that's an impossible question to answer. For a good reason, though, because the biology is always a moving target. Problems continue to evolve and change in Alabama, in the country, and in the world. The issues that are plaguing the field of plant biology, agriculture, crops, pests, pathogens, they keep changing. And actually, they change very, very fast. So as the biology changes, the technology adapts. And as the technology continues to improve, the questions that we're able to answer are also different and changing all the time.
So what is all this?
So HudsonAlpha is a research institute first and foremost. And part of being a researcher, part of being a scientist is that you have to publish your work. You have to let people know what you've done and why it matters, and give some kind of promise towards the future of your work. And so what's really wonderful about the plant group at HudsonAlpha is that every paper that plant faculty have worked on, going back to the first human genome physical map, is up on this wall right now. And so there's this rich history that you can see with your own eyes, not just of the history of how genetics and technology and biotechnology has changed, but also the impact of the kinds of science and the advancements that we're able to make here due to all of this incredible genetic and genomic sequencing technology that we have access to.
Awesome. So one day we'll see your name up there?
Hopefully at the very, very end, yeah. More and more every day.
I think it's really opened things up for us because gene editing is one of those things where, before in plants, we couldn't ask these kinds of questions. We couldn't actually say, that's a gene which we think is involved in this process. Then we have to go look in the diversity collections and look in the germplasm to find genes, find that variance in that specific gene, to then ask that question of, if there are variations in this gene, how does it translate from the gene variation at the DNA level to the gene variation at the RNA level, which is the expression level, to the protein, to function, right? And to do that, we just didn't have the tools. And so now, because you can precisely say, I want to change that specific gene using, CRISPR-Cas9 and gene editing, I can actually now modulate that gene function and ask these questions. And so it reduces the timelines to really discovery. And so therefore, I think it's important as we understand and try to actually discover ways to understand these plants better, to then tell a breeder, for example, say, this gene is really important for this particular thing. You might want to look at the variation in that and bring it in to your breeding program. So that's long term.
Plant biotechnology is advancing the field of agriculture in many ways. Plants have genomes, just like humans do. And using tools like genome sequencing here at HudsonAlpha, we're able to study the genomes of all sorts of diverse plant species, crop plants, for example. And by doing that, by sequencing the genomes of many, many plants, we're able to, again, identify the differences between them. How do some mutations benefit some plants? And Once you figure out the genes that might control a trait, then you can start to select for them in the plant breeding process. So the connection between biotechnology and agriculture is a very concrete one. The more we can do in the field with classical and traditional breeding, the more rapidly we can make gains in a laboratory using genome sequencing. And these two things inform each other very well. You can't do one without the other. Alabama relies on the expertise of its growers and its breeders. But using tools like what we have here at HudsonAlpha with genome sequencing, we hope to be able to accelerate those kinds of gains, to speed up breeding, to allow plants to be selected for faster, to develop markers that segregate with different traits, and ultimately to make more food for the state. But how does this tangibly benefit any farmers or growers or producers in the state of Alabama? One key question that we've been collaborating across many institutions in the state with is a very simple question, actually. Can we bring new crops to Alabama that are traditionally not grown here in high quantity? One really exciting example is a really large collaborative project between HudsonAlpha, Alabama A&M, and Auburn University to bring barley to Alabama. Not traditionally grown in the state of Alabama, but over the last year, through Auburn's variety trial program led by Henry Jordan and Alabama A&M's field stations led by Dr. Ernie Siebert, we've planted nearly 55 varieties of barley at 3 field stations across the state. And maybe a little bit to our surprise, they grew great. And so now we've shown the efficacy, potentially, of bringing a new crop to Alabama. That's step one, though. You have to be able to collaborate with all the field stations and the farmers and the growers to show that you can at least bring diverse plant varieties here and grow them. Step two is our ability to actually sequence the genomes of all these diverse barley lines and start to actually make controlled crosses between individuals and do selections and do marker-assisted breeding. The ability to use genetic information generated here at HudsonAlpha, connecting with the phenotype information generated at the variety trial programs at both Auburn and A&M from growers and farmers and breeders who are literally looking at the plants with their eyeballs, and connecting that information with people like my laboratory and Josh Clevenger's lab and Jeremy Schmutz and Jane Grimwood and Kingshita Swaminathan here at HudsonAlpha, who are using our eyes and computers to look at the DNA of those plants. So ultimately, the goal is to connect all of these groups together, to connect classical expertise in growing plants on Alabama soils, and all this incredible expertise in studying the genetics and the genomics of plants, especially diverse plant species like barley, to improve Alabama agriculture. And hopefully, one day we'll be able to bring a new Alabama-specific variety of barley to the state.